So thank you very much, Professor Roy, for this very generous introduction, and thank you very much, Dean Banavar, for giving me the honor of presenting this Eric Young lecture on a problem which has interested me for a long time, and it's a problem which I think will interest at least some of you, and it's a question which is an old one. It's the question of trying to understand how the various forms that we have in this room, how the forms that you will see if you go outside and look at plants or animals, we heard a talk just now about cavefish, how they come to be. And there's a particularly jaundiced view that I'd like to essentially tell you about, which is what is the role for physics, and what is the role in particular for a very simple kind of physics and geometry, which actually harkens back more than 150 years ago. So many of the things that I want to tell you about could have been, probably should have been done a long time ago, but as luck has it, they weren't, and with a number of collaborators, we've been trying to explore some of the phenomena. I should also tell you a little bit about the title, which was essentially cribbed from this remarkable book on growth and form by a Scottish mathematician and a classical scholar, Darcy Wentworth Thompson. The first edition of this book, for those of you who don't know about it, was published in 1917. So in about 5 years, we will celebrate the centenary of the book. In its first edition, it was more than 1,000 pages long. And as Darcy Thompson himself said, the whole book is a kind of introduction and an invitation for people who are trained in mathematics, trained in physics, trained in engineering, to try and look at biology from a perspective associated with those lenses, associated with geometry. And right in the introduction to this introduction, if you will, he says, and this I will take as a mandate for what I'd like to essentially tell you about, cell and tissue, shell and bone, leaf and flower are so many portions of matter, and it is in obedience to the laws of physics that their particles have been moved, molded, and conformed. Then he goes on to say, the problems of form are, in the first instance, mathematical problems. The problems of growth are essentially physical problems, and so the morphologist is ipso facto a student of physical science. So how, then, can we essentially take this mandate, which he gave us almost 100 years ago, and actually make sense of it? So I want to continue with these quotes and first tell you another one by a giant in biology. It's always nice to start with a quote which is attributed to some of these giants. So Darwin said, when he was looking at diversity of plant form, that it's enough to make the sanest man mad. So if you go outside, you look at the leaves, you look at the flowers, you look at the colors, tremendous diversity, tremendous variety. How, then, can we essentially make sense of this? And here are some images. Okay, so you have leaves which can be very large. Tropical leaves can be meters in size. They can be long. As a function of latitude, as you go from the tropics towards the poles, they become more and more slender. They eventually become needle-like. If you go below water, you see algal blades, which I'll actually come back to. They are not vascularized, so they don't have veins. They don't have networks. They can be very, very large. In one dimension, they can be meters, 5, 6, 10 meters in length. The other dimension, they can be a few centimeters. And the third dimension, they can be millimeters. So from a mathematical point of view, this is wonderful because you have scale separation. So how can we understand these? You look at flowers, and flowers do resemble leaves in some sense. The petals do resemble leaves. Yet they're different because they have color. How can we understand this? That was Darwin's point. A different scientist, more or less slightly afterwards, a few decades afterwards, Rutherford, presumably talking about botany, said, all science is either physics or stamp collecting. Okay? Now, of course, you need to understand the stamps, at least at a level of trying to recognize what the different patterns are, if I want to make sense of what it is that unifies them. And so I'm going to tell you a series of stories which will sound like stamp collecting, because I'm going to tell you about different examples. But perhaps you will also see that as you see the stamps from Hungary, and you see the stamps from India, and you see the stamps from the US, there is a sense in which there is a unity. And that will come because we want to essentially take Darcy Thompson's mandate to seriously. So the kinds of questions one would like to ask from the perspective that I initially came from, but I'm gradually transitioning from the top left to the bottom right, is, are there physical or mathematical models? And by mathematical models, I mean abstractions. By physical models, I mean exactly what Darcy Thompson meant. Basically, can I reconstruct some of those shapes using physical principles, but using physical objects as well? And can those models tell us about analogies between different flowers, between leaves and flowers, between leaves and our gut, as I will tell you about later? And do these abstractions allow us to compress information? Do they allow us to essentially compress information associated with shape, which then naturally leads to the biological question from an evolutionary point of view, what are these unifying principles? And if there are these unifying principles, can I compress information into shape associated with shape into a few different spaces, morphospaces? This is a word which was first coined by David Raup, a paleontologist who was thinking about the shape and the size of shells. Okay? Are there such principles? So that's our goal. You make of what you will if we actually reach the goal, and I'm going to tell you about a few stories along the way. And these stories are associated with a series of problems on different scales and also in different organisms. But there is, I hope you will see, a set of unifying themes. So this is a pollen grain that causes and might cause allergies in the spring. The size of the pollen grain is of the order of a few tens of microns. It can grow a pollen tube. And it's a remarkable feat, which I will tell you a little bit about. So I want to first start with the case of a single cell, but a single cell which is large enough that you can essentially view it with your naked eye, and yet it's capable of doing remarkable things. And the question that I want to ask over there is, how does this tube grow? Because that tube is what takes the male gamete to the female gamete, fertilizes the flower, and as a consequence, eventually we get fruits. I want to then tell you a little bit about how tendrils coil. So think of now taking this to a very large extent. So rather than thinking about a tube, it's a long, elongated object. It's more of a solid rather than something which is hollow. That's an illustration from Darwin's book on the subject. And here is a picture of something that I actually stole from Wikipedia. And I wanted to ask the question of how did the tendril get its shape? So a bit Kipling-esque, if you will. And what can we learn from there? I then want to move from a one-dimensional object to a two-dimensional object. So this is quasi-one-dimensional, if you like. This is truly, or again, quasi-one-dimensional. Then I want to move to a two-dimensional object. How did the leaf get its ripples? And then finally, I want to tell you about, before dinner, how the gut got its shape. Okay? And these problems all have a simple but very beautiful aspect of geometry because they essentially deal with thin objects, filaments, sheets, tubes, and shells. And in fact, you can explain all of them using very old ideas. And again, I want to essentially indicate over here something that we celebrated in this last year, the centenary of Alan Turing's birth. And one of his most famous papers, written just before he died, The Chemical Basis of Morphogenesis, starts with the introduction, which said the theory does not make any new hypotheses. It merely suggests that certain well-known physical laws are sufficient to account for many of the facts. And I hope you will see exactly the same thing over here. Much of what I'm going to essentially tell you about has been well known, and I'm just going to put them together in perhaps somewhat interesting ways to explain some of these phenomena. Okay, so I want to now dive right in and I'll tell you about each one of these at different levels of detail and then see what we will about the principles which essentially connect them. Why are pollen tubes interesting? I already told you pollen tubes are some of the largest cells that you see in the plant world. They can be centimeters in length. They are microns or tens of microns in diameter with a thickness which is about a micron. So again, spectacular scale separation. They start out as a grain, different shapes. And these grains then, when they are on the pistil, which is the female organ, they will essentially grow a tube. And that tube is able to navigate this distance of a few centimeters until it reaches the ovum. Many questions you can ask. For example, how does it know its path? Second question that you could ask, how does it essentially grow so long, so fast? In fact, you can see this with the eye. So I'm showing you over here, this is a video of a growing pollen tube. It's been sped up by just a factor of 4 or 5. So not sped up that much. So visible to the eye, something that perhaps in elementary school, a lot of kids do experiments with today. I mean, I didn't do these experiments when I was growing up in India, but I imagine. And a whole bunch of questions you could ask associated with how does this geometry get set? What is it that essentially allows this object to grow? Because if I want to grow, I have to add matter, but I also want to preserve shape. And how do we account for the fact that I have to think about forces, because there are forces which essentially cause this to grow, and the chemistry associated with how I add new material to the walls in just the right place at just the right time, okay? Again, a very old question. Periodically, people essentially come to this and have come to this, as have we in the last couple of years. I want to make an analogy. Okay, again, in the spirit of Darcy Thompson, the analogy is to glass blowing. All right? So if you have a penchant for beautiful pieces of glass, you ask how they're blown, how they're made. One way to make these beautiful pieces of glass is to blow by heating the glass first in a furnace, taking it out, and then having a tube to which you attach the glass and then blowing. So you have a pressure difference between the inside and the outside. You have a viscosity, the ability of the glass to flow, which is a function of the temperature, and a consequence of the fact that the temperature is non-uniform, and the consequence of the fact that the glass is drooping under its own weight because it's at high temperature and at low viscosity. You use the combination of temperature and gravity with a pressure to essentially create beautiful forms. What's the analog over here? Well, it turns out that the inside of a pollen tube has a lot of molecules, a lot of proteins, a lot of solutes, which essentially means that osmotically it has a higher osmotic stress compared to the outside. So a consequence of that is that the pollen tube essentially wants to bring in water. If now it brings in water, the inside is under pressure. Now if you do the experiment with the balloon, what would happen to a balloon? The balloon will become spherical. So the first surprise is it's not spherical, it's cylindrical, even though it's under pressure. And it's a pressure associated with 10 atmospheres. By the way, that's why your spinach is crunchy. Your spinach is crunchy because the water inside is at a higher stress level, pressure, compared to the outside. And so consequences, when you bite through it, the cell wall breaks. You put the spinach in salt water, you immediately have lost that differential pressure, and you cannot bite through it. It becomes soggy. So exactly in the same sense as in fact, all plant cells have this remarkable fact that they are walled. That wall allows them to essentially sustain a differential pressure. So how does it grow into a cylinder? Well, the answer is simple. The way I would essentially cause things to remain cylindrical is to make the properties inhomogeneous. If it is soft at the tip, but it starts to stiffen or solidify, actually, in this case, as I approach the equator, and I pressurize it, new material will be added to the tip, and that new material will cause things to essentially distend, but only at the tip. So this material that's added at the tip starts to move towards the equator, but a consequence of that is that the tip starts to move. Okay? And what you're seeing over here are vesicles, and these vesicles bring new material to the tip. They lyse at the tip, and they lyse because you have gradients in ion concentrations, in particular associated with calcium. So the vesicles lyse. A consequence of the vesicles lysing is that you have these polysaccharides, large molecular weight sugars, and these sugars then bind to the wall, but the pressure then drives them, and a consequence of that is you get growth. Okay, so that's in words. We'd like to try and convert that explanation in words, which I've essentially illustrated over here using a picture. So I have railroad tracks corresponding to the actin cytoskeleton, along which I have vesicles which are transported. These vesicles come to the tip, they explode over there, they give rise to then this new material, which is pectin, something that you see in your yogurt, that ends up on the wall. If it's softer over here compared to here, or in fact, if it's liquid-like over here and it's solid-like over here, so you have a gelation transition, then this object will start to move, and essentially what you have left behind is a tape recording of the past. So here is the mechanism. New cross-linked, uncross-linked material at the tip arises, flow due to internal turgor, the tip moves forward, and it leaves the tube behind. What are the questions we'd like to ask? Can we use that to predict the shape of this tube? Can we use it to predict the size of the tube? Can we predict the speed of the tube? And can we predict the stability of the tube? Those are the kinds of questions we'd like to ask. And in particular, can we do this as a function, for example, of the difference in pressure between the inside and the outside? Even though you can't control the inside, you can control the outside by playing with salt and sugar concentrations. And can we play with that by playing with the rate at which new material is coming in? For example, by poisoning the inside. All right, so I won't give you any of the details, but I'll essentially tell you about how we've been thinking about this from a mathematical, physical point of view. I should have mentioned this, I forgot, but I think it was evident in the picture, that there is a second mode of growth of cells. The first mode is where I'm adding material mostly to the tip. A second growth mode is where I'm adding material uniformly. Different kinds of cells prefer one or the other. For reasons, again, I'm happy to essentially address at the end. Pollen tubes, some bacteria, fungi, they all grow using tip growth. And if I have tip growth, without having to essentially think a whole lot, you immediately recognize that there must be a length scale where I basically move from adding the maximum amount of wall material near the tip to 0 far away. There's at least one length scale. Okay? That length scale is a transport length scale. there is a second length scale which is telling you how soft the tip is or how fluid the tip is compared to how quickly the fluid essentially switches to a solid. That's a second length scale. So here I've indicated the viscosity, the fluidity of the material, the inverse fluidity of the material associated with the wall as a function of angle. So theta equal to 0, fluidity is maximum, so viscosity is essentially the lowest over there. And as I go towards the equator, the viscosity diverges, because if it didn't, this will become a balloon. And there is one length scale in principle over here. There is a second length scale over here associated with transport. Maximum rate of addition of material at the tip, very little as I move away, going to 0. I'm going to first make the approximation that these two length scales are the same. Okay? So if I want to think about making mathematically progress, assume that these two length scales are the same, and you have to ask, is there a potential balance between the rate at which material is coming in and the rate at which that material is essentially being spread out? Okay, with that assumption, you can actually calculate a steady state shape. So at steady state, so the shape is not changing anymore, it's just moving at constant velocity, and there is just a single parameter which completely describes the system. And that single parameter, again, I'll give you a physical interpretation for, so these equations are eye candy, so you can ignore them. The physical interpretation is associated with saying that there is a transport length scale, which is this parameter A, and then there is a geometric, a material or mechanical length scale R0, which is the size of the tube. And that R0 is essentially a function of the pressure, the viscosity, the rate at which I'm adding material, and so on and so forth. And as a function of just this single parameter, you can see how there is a variation in shape. You can get blunt tubes, you can get sharp, narrow tubes, all as a function of just the single parameter alpha, which is, if you like, a ratio of a transport length scale to a mechanical length scale. Okay, so what can we say about this? So we can immediately ask, can we compare these shapes to what you see in different organisms, all these single cell organisms, fungi, pollen tubes, some bacteria. We haven't really looked at bacteria. I'll tell you about fungi and pollen tubes. Second question you can ask is about stability. And I'll simply mention a word about stability. I don't want to tell you any of the details. So I told you there are two length scales. There's a length scale at which I'm adding material maximally at the tip, and it goes to 0. And there's a length scale associated with fluidity, which says that I'm maximally fluid at the tip, and I become stiffer and stiffer. These two length scales we assume to be different. But in fact, you can't assume to be different, because if you assume them to be different, then I will not have a stable solution. So either the cell will essentially explode, and you can see this. If I reduce the concentration of solutes on the outside or increase them on the inside, the cell will lyse. This is how basically antibiotics work. Antibiotics work because antibiotics essentially monkey with the bacterial cell wall mechanics, and a consequence of that is, again, a large difference in pressure. The cell wall is not maintained, the bacterium explodes. Okay? So the same thing happens over here. And if you want to actually think about stability, we need to essentially have feedback. And this is where we separate from what Darcy Thompson talked about. This analogy to glass blowing, of course, breaks down, because in glass blowing, you control the shape by using feedback. And over here, it's self-regulated. And so the feedback, again, in words, corresponds to saying that wherever you have thinning, if you don't put more material, you're going to run into trouble. And wherever you are thickening, you don't need to put new material. And this is seen in experiments. Again, we can quantify this. And once we do that, we essentially have stability. And that stability criterion corresponds to an inequality between the characteristic length scale of fluidity and the characteristic length scale of transport. You can do comparative morphology, what Darwin essentially asked for, compare different shapes. And you end up with two very simple criteria associated with these general principles. The first one is that the tips are all similar. So if the tips are all similar, you can keep increasing the girth, and it becomes sharper and sharper. The second is where the shapes are similar. The size increases, but the size increases both the radius and the tip radius increase. And you actually see both these in nature, and that comes out of this simple theory. Pollen tubes have similar shapes, so the actual size doesn't matter. And the reason pollen tubes have similar shapes is because the machinery associated with the actin increases as you increase the size, so you're able to keep up. While in fungal hyphae, you in fact have something called a Spitz incorpor, which essentially tells you that a certain amount of material is all you can get per unit time, which means that the tip radius remains the same. And the consequence is that you go from something which is blunt to something which is sharp as you change a single parameter. And you actually see that this seems to be the beginnings of an organizing principle for what determines the shape of different tip-growing organisms. So what is plotted over here are different fungi. They don't always have the same slope. So this is one fungus, two fungus, the third and a fourth one. And then what is plotted over here is a pollen tube. If the size of the tube compared to the tip size is the same, that means the shape is invariant. And so pollen tubes lie along this, while fungi lie along that. So this is the beginnings of using very simple physical principles. In particular, the balance between growth and mechanics to understand how geometry arises. That's my first problem. My second problem is to think about how, and tell you about how tendrils coil and unwind. Okay, so I want to show you a movie first. This is now time-lapse. So this occurs over a few hours. This is a wild cucumber plant. And the wild cucumber plant will essentially have tendrils, which start out as straight shoots, green shoots, deliberately placed close to a bamboo pole. Eventually, they interact with that bamboo pole, and then something beautiful happens. And this is a remarkable adaptation in the plant world, especially found in the tropics. So if I want to grow tall, why do I want to grow tall? I want to grow tall because I can then soak up sunlight. So photosynthetic efficiency becomes high because I can have my plants as high as possible. I have two ways to do that. I can build my own structures, or I can be a parasite. And in order to become a parasite, what I have to do is I can work very well in the tropics because I have plenty of other trees, and I can essentially create these shoots which wind around, and I can hoist myself up to the top of the canopy. So you see this adaptation again and again in the tropical world, where you start, so I think I've played this movie, movie's playing along, you see the shoot, it hooks up with a bamboo pole, and then it starts to coil. And what you can also see is that one part of it has one chirality, and the other part has a reverse chirality. Okay, so this part is a helix which is one-handed, or say it's left-handed over here, and that part is right-handed. This is a necessity. In fact, Darwin already thought about this. Darwin wrote a whole book on the climbing movements of plants, and he said, correctly, he recognized immediately why this is happening. So what's happening is that the plant shoot is growing, the tendril grows, and it attaches itself. Once it's attached itself to the shoot, to the bamboo pole, the total amount of twist, which is essentially initially 0, has to remain 0 because you can't untwist it at one end and you can't untwist it at the other end. One end is essentially clamped to the pole and the other end is attached to the tree. Now, through a mechanism which I'll explain in a minute, I have to have, if I wanted to twist up, the only way I can do that is continuously twist on one side, on one half, and twist on the other side, the opposite chirality. And you can actually see this with these things which now are becoming extinct, telephone cards. Okay? So telephone card has a single chirality, but you often end up twisting the cord every time you essentially switch your telephone cord from one ear to the other, especially if you have the habit of doing it this way. And when you do that, you can essentially develop a kink. And I'm sorry that people far away can't see it, but now, so if I open this up, at least people in the front can see this, I have a helix of one chirality on one part and a helix of the opposite chirality on the other part, and then I have something which mathematicians, topologists have called a perversion, a mathematical perversion. And this mathematical perversion has to be present purely because of topological reasons. Okay? So that's what you actually have. Again, an old problem, and again, there have been attempts over various eras, including the most recent one, where people have posited that it arises due to growth. Because after all, if I have a long rod or a long filament, and if one side grows compared to the other, it's going to bend. So the question was, is this really true? First question, is that growth model really true? And the only way to find out is to go to the lab and measure. So I'm going to tell you about some experiments. What is the mechanism of coiling? What is the origin of the perversion? I already told you. And what are the functional consequences? And that last one is particularly interesting. So here are a series of images of what happens to the tendril as a function of time. So initially, it's straight. This is a cross-section. It's not circular. It looks like a kidney bean. And what you see over here is nothing, and you're expected to see nothing, because this is essentially the autofluorescence associated with the fact that the tendril, as it ages, becomes more and more woody. Okay? And wood is essentially cellulose plus molecules which cross-link it strongly and give rise to something called lignin. Okay? And what you see with time, as the tendril starts to coil up, the cross-section remains more or less the same, no surprises there, but you start to see autofluorescence on one side. Okay? So you start to see a lignification or the beginning of wood, the formation of wood on one side. So it's not driven by growth, it's driven by the fact that the properties are changing on one side compared to the other. If you essentially are very careful, and these experiments are done by Josh Pusey, very careful and peel this tendril so that you can essentially get a handle on this dark blue fiber, you find that the fiber reflects the geometry of the tendril. In other words, the tendril is simply slaved to the geometry of the fiber. The fiber is very stiff, the tendril is very soft, so whatever the fiber does, the tendril does. Okay, so we are starting to understand what's going on. What's going on is associated with the formation of this woody area, and this woody area by definite, or by this theorem, which I think goes back at least to listing, says that if one end is fixed and the other end is also fixed, I must have at least one perversion or an odd number of perversions. All right, what's the mechanism of coiling? So the mechanism of coiling is associated with what you see when you leave a paperback in your bathroom for too long. So if you leave your paperback in the bathroom for too long, then the cover coils, the cover folds up. Why does it fold up? So paper is actually cellulose, the same material that this is. And paper is hygroscopic, so it absorbs moisture, but the cover is typically coated with cellophane, something which is impermeable. So a consequence of that is that the inside of the cover absorbs moisture, the outside does not, and so the paperback curls up. It's the same mechanism that nature has, and of course humans, being humans, have rediscovered it much later. So if I take out the fiber, that's the gel fiber. It's a hygromorph. It's essentially changing its shape because of the presence of humidity. And you can see it starts to coil up or uncoil as a function of the environmental humidity. And you can see and do the same thing with the whole tendril. When you dehydrate it, you see it coiling up. And then when you rehydrate it, it opens. So it's a water-driven engine, and plants are the great masters of water on our planet. They can move water up a giant sequoia, and you never even hear it. Okay? I've already told you about the perversion, so I've told you about the mechanism. It's essentially associated with using water very cleverly to change its shape in the presence of this woody fiber. The perversion is a topological necessity. What is the functional consequence? So Darwin, in his book on the climbing movements of plants, said the tendril strikes an object, curls around, contracts into a spire, dragging up the stem, and forms an excellent spring. So I'd like to ask, what kind of spring does it form? And you can do measurements, and you can do experiments. These were done by Sharon Gerbode. So here is one of these coils with the perversion that I just told you about. And if you do this, the easiest way to do this with a piece of rubber is to essentially take the same piece of rubber, or two different pieces of rubber, stretch one, glue it onto the other one, release it. Okay? Just like what happens when you essentially have something which is woody. You pull the ends apart, and the whole thing unwinds. No surprises. But if you do the same thing with the tendril, it does not unwind, it overwinds. So if I start with 5 coils on one side and 7 on the other of opposite chirality, and I pull on it, I go from 6 and 8. So it's doing exactly the opposite of what a normal piece of 2 rubbers glued together does. And of course, the question then is why? Younger tendrils behave like this, older tendrils overwind. This also, by the way, is seen in DNA for somewhat different reasons. All right, so I want to essentially cut to the chase, and the reason that this happens is that long objects like this filament are very, very easy to bend, very easy to twist, very hard to stretch. All right? But there is one parameter which you actually have control over when you think about the properties, and that one parameter is the ratio of how easy it is to bend compared to how easy it is to twist, forgetting about the other modes completely. And if you vary that parameter, and just that parameter, and this is associated with the fact that you lignify. The fact that you lignify means that you have a single parameter, which is the ratio, if you like, of the bending stiffness to the twisting stiffness, that changes. And if that changes, you can show mathematically, you can solve the problem numerically and semi-analytically, you essentially see that when that parameter becomes large, you see exactly what you see in nature. Namely, that the fact that it's lignified now makes it overwind. Of course, it can't overwind forever. It initially overwinds and eventually it unwinds. All right, so what's the consequence of that? the functional consequence of that, these two facts, one, that I have a perversion, and two, that it overwinds, is beautiful. It actually makes for a spring which is very soft initially. And the reason it's very soft is that I have this perversion which essentially acts like a place where I can spin. And the second part, later on, it starts to overwind, means that it stiffens. So it's initially soft and eventually stiff. And I'm going to now indulge in a little bit of teleology, because this suggests that these tendrils do exactly what they should be doing, because initially the plant hasn't still figured out where it wants to essentially have a structure. So you want something which is soft, but once it ages, you want it to start to become stiffer. So I apologize for the teleology, but that's an engineering principle that you can uncover, and you can essentially check that against experiments. You can check that with a calculation, and it leads to a very sort of naive but yet useful generalization or a variant of the humble helical spring, which has been used forever, because you essentially introduce this perversion and you play with this parameter by making it woody on one side, and you get something which is initially soft and eventually stiff compared to the normal helical spring. Okay. I told you about a tube. I told you about a one-dimensional object. Now I want to move to two-dimensional objects. And I want to tell you about algal blades. And the reason I want to tell you about algal blades are because marine plants have a very nice, much nicer time than terrestrial plants for two reasons. One, they don't have to support their own weight. Terrestrial plants have to support their own weight, and this is a huge issue. Marine plants don't. The second is that terrestrial plants have to essentially pull all the nutrients that they need from the soil, while marine plants essentially get them from the environment. A consequence of that morphologically is that marine plants are not vascularized, so they don't have beautiful venation patterns. You don't see veins in marine plants, in the algal blades and so on. And that makes life simple mathematically. Okay? It's interesting biologically, but it also makes life simple mathematically. And what I want to tell you about are some experiments which I was introduced to by Mimi Cole, who actually did these off the West Coast near Friday Harbor. Not Friday Harbor. Yes, Friday Harbor Labs near Washington, near Seattle. And these experiments show that there is an enormous plasticity of form. Form is the phenotype that you're interested in this particular algal blade, but it's actually generic, in Neurocystis leukina. So algal blades are very long, meters, wide about 20 centimeters, and thickness is about a millimeter or two. They're approximately neutrally buoyant. All right? So they're more or less vertical. But now there's a problem. And the problem is that they might get into each other's shadow. So how do they essentially solve? Again, I apologize for the teleology. I'm happy to essentially talk about this at length later. So what they do in slowly flowing environments is that they end up with blades which are ruffled along the edges. You take this algal blade, Mimi Cole takes this algal blade. So I should also tell you that growth is not uniform. Again, this is a tape recording of the history of the algal blade. This was what was developed long ago. This is more recent, and this is now. And growth is not uniform. Growth occurs primarily at the place where the blade is attached to a support. In slowly flowing environment, the edges are rippled. If the edges are rippled, even at slow flows, it will start to essentially oscillate. It will start to flutter. And if it starts to flutter, then you have a reasonably high photosynthetic efficiency compared to a blade which was flat. So from a selection point of view, from an evolutionary point of view, these guys will essentially outgun because they can grow more, they can grow faster than anything that we cannot. But there is a competition. The competition is associated with what would happen if I switched my environment. So Mimi Cole transplanted these algal blades from a slow-flowing environment to a rapidly flowing environment. And you can do this over one season 3 or 4 times. The new material that's added, the old material is there. The new material that's added is now added so that you essentially get narrow, straight blades. Okay? So that's the plasticity associated with the phenotype, which is driven from the environment, by the environment. You change the environment, the morphology responds. Why is this interesting? This is interesting because, of course, at high flows, you have enough fluid dynamical pressure, forces, which essentially cause it to flap. And furthermore, if we had to compare this with this at high flow rates, this would have a selective advantage because this would essentially get ripped off because the drag is high. Okay? And you can do this experiment again and again. In one season, 4 times. Go back and forth, and it will switch. And this is environmental control of morphology. You can make this quantitative by essentially taking a blade. So if this is my blade, I punch holes along the center and I punch holes along the edge. And I just track the distance between the holes as a function of time. And what you find, no surprise, is that here the edges are growing more than the center, while over here they're not. Okay? So that makes perfect sense, because I have to take a flat sheet of paper, and if I wet the edges have swollen. If the edges have swollen, then they start to buckle, while the center remains flat. So the mechanism by which this is generated, top down, is simply that the edges are growing compared to the center, while over here they both are growing more or less at the same rate. The solid corresponds to the center, the unfilled corresponds to the edges, while over here, the unfilled is growing much more. And furthermore, so this is the growth rate as a function of location, and you have very little growth here, all the growth is limited over there. So we understand what's going on, all right, qualitatively. You can also do experiments bottom up using genetics, okay? There's a beautiful paper published about a decade ago called The Genetics of Geometry, asking the question, how can I essentially try and control the shape, or can I control the shape, in this case, of the antirrhinum, the snapdragon leaf? Already, this should essentially force you to think about a beautiful problem that nature has solved, which is to grow flat leaves. There's only one way in which a leaf can grow flat, and an infinite number of ways in which essentially it can grow non-flat. I won't address that question. I will leave that to the end. But what these people did, Enrico Cohen and his group at the John Innes Center in Norwich, is that they were able to get a gene expressed associated with cell proliferation called the Cincinnati mutant, which focus on these two pictures. What's being shown over here is the rate at which cells are proliferating on the side compared to the rate at which they're proliferating on the center. What you see is that the front, which corresponds to the fact that there is a propagation of cell proliferation, is more or less uniform. If anything, the edges are growing slightly less than the center. If the edges are growing slightly less than the center from a flat plate, it'll become a bowl. But if the edges are growing more along the edge, the edges are growing more than the center, it will become a potato chip. By the way, that's why a potato chip has the shape that it does. Also, if you take a potato chip and you put it in boiling oil, water first leaves from the side. That's because of a beautiful analogy between that problem and the field in a capacitor, field in a thin conductor in a vacuum. There's a singularity, and exactly the same thing happens over here. And the consequence of having more growth along the edges gives you ripples. Okay, so you have bottom-up and top-down control. You can also do experiments yourself. Nobody complains if you essentially do surgery on plants. So here is a leaf from the Asian lily, which has a beautiful saddle shape, but also has ripples along the edges. You cut away the midrib. The midrib springs back. You cut parallel lines to the midrib, and all those lines, all those strips essentially straighten out. Okay? Saying what? Saying that the morphology is a consequence, I've told you this already a few times, is associated with the fact that the edges are growing more than the center, but just as importantly, it's then a consequence of just minimizing the elastic energy. Unlike the problem associated with the pollen tube, where you have to add material and flow, over here, it's a consequence really of minimization of energy, as these experiments suggest. And if you follow Darcy Thompson, you can essentially also repeat the experiment with a piece of foam. You pull your nails along the edge, and you pull your nails along the edge will stretch more than the center. You get a saddle, and if you pull it substantially, then you also will get ripples. Okay, so that's the physics and a little bit of the biology. There is a bit of mathematics, which I'm going to spare you. I'm going to simply tell you the results of some numerical calculations, which more or less repeat the analysis, but can be couched and moved into a strongly nonlinear regime. You can basically try and construct a model of this leaf where it's very long and described by a lattice of springs. And these springs are such that it costs you energy to stretch them in the plane, but it also costs you energy if two triangles which are next to each other are non-parallel. Their normals are non-parallel. Okay, so I have two kinds of energies, a stretching and a shearing energy and a bending energy. Where is the biology? The biology comes in and saying that the edge length is not its rest length. So the consequence of that is that the edges are longer, this is going to ripple, and out comes a morphospace. So morphospace is essentially characterizing the shape of these blades as a function of the only parameters that remain. Parameter 1 is the aspect of the blade. It's very long, so the length doesn't matter, but I'm looking at the aspect comparing the width to the thickness. That's this axis. And the horizontal axis is how much I have grown along the edge compared to the center. So if I'm a very narrow blade, so W is small, if beta is small, it remains flat. I have to essentially make the blade wider and wider in order for the edges not to talk to each other before it switches from the straight to the saddle, flat to the saddle. While if I wanted to increase the strain, it essentially means I'm going to move in the horizontal direction. As the edges grow more and more, for whatever width I have, I switch from something which is straight to something which is saddled, and eventually from something which is saddled to something which is rippled. And there are calculations that you can do which tell you where the boundaries between these different phases are. which says that what the plant has to do, which I think I've already told you multiple times, but it's worth emphasizing now in the context of the plant, in morphospace, is the plant is essentially fiddling either with the aspect going this way or with the relative growth. And either one of these, if they respond to the environment or bottom-up or controlled by genetics, allow you to change shape. Okay, so I've told you about tubes, I've told you about strings, I've told you about plates. I want to now combine them. I want to combine them in a completely different context by asking how your gut got to where it is. So your gut is a few meters in length, 7.5, 8 meters in length. It's packed into a cavity. Hopefully the size of the cavity doesn't grow with time. Okay? And I want to actually show you some images of what happens not with the human, but with the chick. And these are experiments done in Cliff Tabin's group at the medical school. So they are geneticists, and they're interested in trying to understand, among other things, the genetic basis, not of geometry, but the genetic basis of chirality and broken symmetry. Our lungs are not symmetric. Our liver is on one side. Our heart is on another side. And so they ask, how far back can you essentially go and say something about when this happened? During this process, they also started asking about how organs can or cannot essentially lose their chirality. And they started thinking about the gut a few years ago when I first met them. And so let me actually introduce you to the problem and the question. So this is the chick gut 3 days after fertilization. And to orient you, so this is the brain or the forebrain. That's the vertebral column. This is the tail. And this herniated object is the gut. Okay? So the size of the gut 3 days after fertilization, this object is about 7 millimeters long. Now I have to now tell you something about how the gut is essentially formed. So the gut is a tube. And the reason they were interested, the reason I got interested, is because the gut is a very simple object. It's a long tube, but it's a very long tube. So the question is, how does it essentially fit itself inside the body? So the gut is attached to 2 holes at either side, and it also is attached to the body along itself by a piece of tissue called the mesentery. All right? So the question is, does the gut keep herniating out? And the answer is no, because it has to get packed. So it has to essentially flip on one side or the other. So they asked the question how that happens. And they discovered that there is a particular gene called PITX2. And that is when it's essentially the proteins corresponding to that are expressed, it causes differential proliferation. Just like what I told you about the leaf. On one side, so this is the tube. This is the fat mesentery, which I'll show you images of in a minute. One side of the mesentery grows relative to the other. A consequence of that is that the gut starts to twist on one side compared to the other. All right, so they said, okay, so this is great. So I think we understand the onset of the chiral behavior or the beginnings of chiral behavior, but I already told you about the tendril problem. And the tendril problem, the ends are fixed. So even though it looks chiral, it can't be globally chiral, because if it does something on one side, it has to cancel it on the other side. So topology has to be preserved. So what happens later? That's day 3. This is day 5. You start to see the formation of a helix. This is day 8. You start to see something which actually is slightly more complicated. Day 12 and day 16. So I should essentially tell you about comparative times. The chick hatches at day 21. All right? And this is the gut from one chick. This is the gut from another chick. And these look very similar. So it seems like there is something interesting over here, not only at the level of the first breaking of symmetry, but what happens eventually. This is a magnified view of one of these. And what you see are beautiful loops. Okay? Very reproducible. How did they come to be? Well, if you were a geneticist and you discovered that one side was essentially proliferating compared to the other, you would say, well, maybe it's the same mechanism all the way through. Every time I wanted to form a loop, I grow the top compared to the bottom, I grow the bottom over here compared to the top, and so on and so forth. That's one possibility. Another possibility is to say, well, it looks reproducible, but there is some variation. Maybe it's a solution of this packing problem that I told you about earlier, because I have to pack all this inside the body cavity. Let me start with that hypothesis. How do you essentially corroborate or destroy it? Cut. Okay? You cut the gut away from all the anchors, and that pattern persists. That red thing over here is the supramesenteric artery. So, and that you see over here as well. So the loops persist. So it is not a solution to a packing problem. Okay? So then I want to come back to the other possible hypothesis. And the other possible hypothesis is the master puppeteer hypothesis. The master puppeteer hypothesis says that every time I want to essentially bend, I grow on one side compared to the other. But then I have to exquisitely orchestrate that, and that's what the puppeteer is. But genetics can do amazing things, and perhaps that's what's happening. Okay? So here are images of the gut. And the only reason I want to show you is to make one important point, which I made earlier, but it's very, very visible over here. So this is the tube. The chick you can't see, which is on the next floor. And one side is growing compared to the other. That's the original broken symmetry. This is at E16. There's the gut. And you see that mesentery, which was a thick tissue, has become a membranous, diaphanous, very thin membrane. Okay? And during this process, you see all the coiling. So what we can do in order to ask the question, is this the master puppeteer hypothesis? We can label the tissue and ask whether one side is essentially proliferating more than the other, whether there are more cells on one side compared to the other, at different locations along the gut, on the side, on either side, and then also ask whether there is differential proliferation along this compared to that. And I'll just run through the results. There is no variation. So if I essentially look at how the gut proliferation, so how cells proliferate or divide as a function of the azimuth, they essentially remain the same, number of the cells dividing. If I do the same thing along the mesentery, along this, right here, where the gut is attached to the sheet, you also see the same, no variation at all. But if you look at the numbers, you see that the number of cells dividing along the mesentery, right there, compared to the number of cells in the gut, you see are off by a factor of almost 10. Which means that the membrane which connected the tube to me is actually growing less than the tube. How do you verify that? Surgery again. This is the gut cut away from all the anchors, and now you tear away the mesentery, that diaphanous membrane. It collapses to this, and the gut's completely straightened out. So it's the same principle that you see in the leaf, but now in a completely different system. It's also very elegant because it says genetics is important only in determining that you have a differential proliferation, and then stupid physics just takes over. Okay? All right? So can you essentially make a model of this? I'm almost out of time, and the answer is yes. You take rubber sheets, you take rubber tubes, they cost a few cents each. The tissue, the mesentery, the membrane is the latex sheet, the gut is a rubber tube. You essentially stretch the mesentery, the sheet, attach it to the tube, and shrink and let it relax. So that's differential growth, because the gut is put into compression, the sheet is put into tension. Okay? And very important, I can't forget the topological constraint, because the ends are fixed. This is the gut of the chick. The only new thing that's been done is that supramesenteric artery has been cut. Okay? And you see the shape over here, and proof of principle. Here is the shape of a rubber tube on a completely different scale, completely recapitulates what happens. Okay, so we understand what's going on. In the Darcy-Thompson-esque view, you've essentially got a physical model. And then when you have a hammer in your hand, the whole world looks like a nail. And I was walking around the street in Paris a year or so ago, and I saw a boutique associated with this person, and she sells these as fashion items. But they cost, instead of a few cents, they cost a few thousand euros. So I have to orient you. So this is the neck of a mannequin. And what she's done, what Susan Ehrman has done, is take diaphanous sheets and attached with a hem a very much stretched or compressed piece of wire. And a consequence of that is that you reproduce the pattern that she probably doesn't realize is in her gut as well. Okay? All right, so you get greedy. So you can do the mathematics, or you can do the physics. You can start to think about the biology. You want to now build a mathematical model, which includes the geometry, which includes the mechanical properties, and of course includes relative growth. I'm almost out of time. You can do a little calculation. You can do a numerical calculation and an analytic calculation. An analytic calculation for the size and the wavelength of those loops and the amplitude of those loops in terms of things, all of which we can measure. The elasticity of the gut, the geometry of the gut, the radius of the tube and the thickness of the tube, the elasticity of the mesentery and the thickness of the mesentery, and also the relative growth. Okay? So I won't tell you how we got those results, but you can essentially do that. You can do a numerical calculation, and you won't be surprised if I tell you, of course, you recover this. But the more interesting thing is to compare the egg with the rubber, with the computer, with this little analytic calculation. No parameters. Everything was measured, okay? And so these are the purple correspond to experiments done on a computer, the easiest, the cheapest. So you can see that they span substantial range. Black is different stages in the chick, E8, E12, E16. Green are the experiments with rubber. And as I said, without any parameters, we can get this prefactor of 36 by doing a numerical calculation. The scaling arguments are one line each. You get both the wavelength and the amplitude. Okay, so it doesn't do so badly. And as I said, you get greedier and greedier. So then you ask, well, does this work in other organisms? All right, a little aside. So auspice and haruspice was practiced by the ancient Etruscans and Romans and Greeks because they wanted to try and predict the future by looking at the entrails of birds and sheep and so on. We haven't succeeded yet. Okay? But in the last century, or actually I should say the 19th century, there's been some spectacular morphology associated with bird guts. Why? Just like bird beaks, like Darwin's finches, who we think have essentially a fantastic selective advantage associated with the kinds of food they eat, bird guts are very interesting because of the diet they consume. Okay? So if I consume a carnivorous diet, it pays for me to essentially have a short gut. While if I have a herbivorous diet, I want something which is long so that I can essentially absorb all the nutrients that I possibly can. In fact, there are animals, voles and other rodents, where in one organism, if I essentially change the diet, the gut length changes. So there is that much plasticity. All right? So we wanted to ask, can we essentially make sense of the principles, not in terms of predicting the future, but at least predicting the past? What had happened in the embryo in order to develop these complex patterns? And we did experiments to essentially compare the chick, which I told you about, the quail. Why the quail? Well, the quail because the chick egg is almost a factor of 10 or larger in volume than a quail egg. But shape is shape. Who cares about size over here? Because I'm telling you about shape. You look at the finch, which is a favorite of evolutionary biologists, and then you challenge the genetics. But I've actually told you, genetics plays no role whatsoever, and you go from the avian to the mammalian. You look at a mouse. And for each one of these, you measure the geometry, the size of the tube, the length of the tube, the relative growth of the tube compared to the membrane. You measure the mechanical properties, and you plot everything. And you see that, again, there is a reasonable agreement, again, with no prefactors, with no fudges. Everything is measured, and you just stick it into the little formula that I showed you a slide ago. Okay, so I am out of time, and I want to stop here. So we are continuing to think about what happens later in gut morphogenesis, because the gut doesn't remain a uniform tube. The gut eventually develops finger-like protrusions, which are villi, and these villi, of course, are associated with absorption of nutrients. They increase the surface area. How do the villi form? And it turns out until almost the last stage, the villi form, again, using the same principle, which Darcy Thompson already elucidated. Now, the inside of the tube grows relative to the outside of the tube. First, azimuthally. So if I now have a ring inside another ring, and the inside ring grows relative to the outside, it will essentially form corrugations. And then it varies along its length. And those corrugations then become more or less finger-like. So the same principle is being used again and again. It's an old idea. All these calculations could have been done a very long time ago. We just got lucky. And I'd like to close by going back to Darcy Thompson, who said, again, in that book that I told you about, that the zoologist and the morphologist have been slow, but the physiologist has been eager, long, to invoke the aid of physical and mathematical sciences. With quantitative experiments, which in principle could have been done a long time ago, just measuring simple things, we are starting to get at a physical basis and a geometrical basis for morphogenesis. And of course, the most interesting aspect of this is to try and compress all this diversity in shape. And shape, in a sense, is a prelude to function. Anatomy is a prelude to physiology. And so if we can understand shape, can we then start to ask questions associated with function as well? I've told you only one half of the story. In any interdisciplinary area, one hopes that you essentially have things which go both ways. I've shown you that very simple ideas from geometry and mechanics essentially lead to interesting ways to think about biological problems. But in fact, in many of these problems, and I unfortunately don't have the time, it also goes backwards, that you have very, very nice questions which essentially are interesting from a mathematical point of view, which combine geometry and analysis. For example, the sheet, the leaf, is essentially, once it starts to grow, is a non-Euclidean surface which is being embedded in Euclidean three-dimensional space. What are the optimal ways of doing this? If I use elastic energy as a criterion, I can start to get there. There are beautiful questions associated with describing variations in shape. I want to look at my gut and I want to look at somebody else's gut, or my eyes and somebody else's eyes. How do I understand the coupling between statistics and geometry in order to essentially determine where there is a difference? What does it mean? And I've already told you from a biological point of view that this very simple idea is differential growth. I give you one and only one example of regulation. The first one, where I said there's feedback. We are starting to think about how regulation comes in over here, because, of course, that's what separates the inanimate, in some sense, from the animate. I didn't tell you the details, but what we find in all these organisms where we looked at the development of morphology, the final strain is exactly the same. So rather than, as Turing himself already pointed out, although he didn't focus on it, he said that there is essentially a coupling between the reactions and the diffusive processes. If there is a coupling between relative growth, which gives rise to shape, but shape then gives rise to stress, and then stress feeds back on growth, you're done. I told you in the context of the pollen tube, we're getting there. In these problems, not clear. So that should keep us busy for some time. Thank you very much. I should say, yes.
What place does chaotic behavior come into effect? What do you say about chaotic behavior?
There is no chaos. It's very regulated, and it seems to be extremely regular. I mean, if I think of it in the literal or mathematical sense, I'm not sure whether there was a specific aspect that you were actually or you meant to focus on.
Is there any challenges that an organism faces where a chaotic response would be to its benefit?
In terms of morphology, nothing that I can immediately envisage. Self-similarity and things of that sort, where you have all kinds of structures on multiple scales, very naturally comes up. Leaf edges, the edge of your lattice, for example, over a certain range, looks like it's self-similar. So you do have multiple spatial scales, but I'm thinking about multiple time scales. I don't see immediately. I mean, because many of these structures evolve very, very slowly over the period of development in embryogenesis. So,
thank you. So you talked about the monogastric gut, which is pretty monotonous. Same kind of intestine. But if you see a ruminant animal like a cow's gut, it has 4 parts. One is like a beehive. One is like a leaf. One is like the other part is mostly like humans. So how do you explain the formation of those beehives and?
I don't know. Yeah, so that happens later. Okay, so the short answer is I don't know. There are places where we are starting to think about it, not in the context of gut morphogenesis, but in lung morphogenesis, where you actually do see branching. You do see the formation of different types of compartments, but in the context of the gut, I just don't know.
So thank you for a wonderful lecture. You explain this more from a mechanical point of view, how these
Indeed.
things happen. There was a book like about 8 to 10 years back on cellular automaton, and there the argument is taken more in a simple rules-based development. So is there any connection? Because they simply Stephen Wolfram simply talks about simple rules that cells decide on based on whatever their internal structure and their biology, and they just continue to follow those rules, and you get all different shapes. And different
Who decides the rules?
Yeah, so that's what
I'm not using anything that's new over here. I'm just using stuff that nobody will disagree with. I'm just using the fact that things are growing, and they're not growing uniformly, and you can explain it. And I think there is an economy which we all, I think, would like when we're trying to explain something. If I have complex rules and I get complex output, I mean, I don't know. So I don't have anything to say besides that. The fact is that I took stuff which Rayleigh and Kelvin and Stokes knew, and it turns out that it works. So I have nothing to say to that besides the fact that it works. So.
There's some interesting questions about gut physiology.
Yes.
In terms of like meat digestion versus vegetarian
Yes.
digestion.
Yes.
And the fact that we've evolved, we need meat for high metabolic, blah, blah, blah. But is it, is there anything known about whether you can reverse the insides, if you become a vegetarian or...
With humans, no, but as I was mentioning, with rodents in particular, voles are a great example of this. You change the diet of a vole from a carnivorous to a herbivorous diet, the gut length will increase. You reverse, and the gut length will decrease. You see this in certain kinds of frogs, and there's a particular chemical, retinoic acid, which essentially is associated with playing with cell proliferation rates. So the short answer is yes, in some organisms. It's probably not true in us. But then it also begs the question, is it because we don't have the ability to essentially synthesize this? And can you then use this as an engineering therapy? And I don't know the answer to that.
Go ahead.
I'll add a little bit to that. So as children and as infants and even inside the womb, we have a spectacular ability to regenerate. Okay, so we eventually start to lose that. So an interesting question that I think you are eventually probably headed towards is, how should we think about this gradual loss of plasticity, which is there in us, and it doesn't occur in us globally. It occurs in us actually at different parts of the body, different rates. I've been talking to a surgeon, for example, who's essentially able to reconstruct an esophagus. So there are children who are born, they are viable in the mother's womb because they are being fed by the placenta, but the children are born without a connection between the throat and the stomach. They're not viable. This surgeon, he does puppetry. What he does is he cuts open the back, and then he essentially attaches with fish hooks. He attaches the top of the protoesophagus and the nub on the stomach, and he pulls them over 4 weeks, keeping the child in general anesthesia. In 4 weeks, you can essentially grow that tube by an amount which is a few centimeters, cuts the ends off which are scarred, stitches them, they never come back. So the point is that there is an incredible physical plasticity and a feedback. I haven't really had a chance to talk about this because we are just beginning to ask this question of regulation. I mean, the simpler question is growth or relative growth leads to shape, shape leads to stress, and it back regulates. And by playing with that homeostasis or lack thereof, by making it imbalanced, maybe we have ways.
Any other questions?